In this video, we'll discuss about the development of active and passive immunity. First, what is passive or active immunity? Passive immunity is when a person is immune towards a certain type of disease due to them being given antibodies to the disease rather than producing the antibodies themselves. The antibodies was produced by someone else's immune system and not their own immune system. There are two types of passive immunity: artificial passive immunity or natural passive immunity. The artificial passive immunity refers to injection of serum with active antibodies taken from other individuals who are already immune to that particular antigen. For natural passive immunity, it involves the transfer of maternal antibodies to their child via the placenta or the breast milk. Either way, both natural or artificial passive immunity is short-lived. Means that the protective value of the antibody will not last long. Since the antibody was not produced inside the body of the person, this means that their own immune system has not been activated. So, there are no memory B cell or memory T cell present inside the body of the person receiving the passive immunity. Without memory B cell or T cell, the person is still susceptible to future infection. Unlike passive immunity, for active immunity, it involves activation of the adaptive immune system. This is why exposure to antigen is crucial in the active immunity. In natural active immunity, the exposure of antigen happens naturally during infection. However, for artificial active immunity, exposure to the antigen happens because of vaccination. Either way, both types of immunity tend to be long-lasting, since activation of adaptive immune response will lead to production of memory B cell and memory T cell. These memory cells will act as immunological memories that can protect the body against future infection for the same antigen. Here are the key differences between passive and active immunity. As mentioned previously, active immunity can be achieved through vaccination. But first, what is vaccine? Vaccines are suspension containing killed, weakened, modified version of the pathogen or antigen. Injection of vaccine into a person will introduce the antigen to the immune system. This then leads to activation of the adaptive immune response against the pathogen or antigen. As a result, the body will produce antibodies and effector cells that can fight the pathogen should the natural infection take place in the future. This shows that vaccination is an effective way of protecting people against harmful diseases before they came into contact with the diseases themselves. Since vaccines only contain the weakened, killed, or modified version of the pathogen, it is incapable of causing disease. This means that we will not get infection from the pathogen present inside the vaccine. Here are some examples of vaccines currently available to the masses. Next, we will look at the characteristic of immune response during the first time a person is exposed to the antigen in comparison to the subsequent encounter with the same antigen. These are what we call primary and secondary immune response. The primary immune response is the reaction of the immune system during first exposure to the antigen. In contrast, the secondary immune response is the reaction of the immune systems during subsequent exposure to the same antigen. We characterize the primary and secondary immune response mainly in terms of the concentration of antibodies produced against the antigen. Here we can see that The concentration of antibody produced during the primary immune response is much lower compared to the concentration of antibody produced in the secondary immune response. As mentioned previously, the primary response is the immune response after initial exposure or after the first time our body encountered the antigen, while the secondary response takes place after secondary or subsequent exposure to the same antigen. During primary response, after exposure to a specific antigen, for example, in this case antigen A, for the first time, there is a lag of several days before the specific antibody becomes detectable inside the body. This means that the first time our body encountered a particular antigen, the immune system is not capable of immediately producing antibody against the antigen. Why? This is because the adaptive immune system is not activated yet. The process of activation will only start when the immune system is exposed to the antigen. And the process of activation will take up several days that we call the lag time. Once activated, the B cell can now proliferate and differentiate into plasma cell and memory B cell. Plasma cell will produce the antibody against antigen A. It is because of the need for activation that the primary immune response have a slower response time. Next, the class of antibody primarily produced during the primary immune response is class IgM. IgM is short-lived, means that it will not last long inside the body. Eventually, the concentration of IgM will decline, means that the protective value of primary immune response is only for a short period of time. Next, if the person got infected by the same antigen, which is antigen A, you can see that the concentration of the antibody rises rapidly and in greater amount in comparison to the primary immune response. In addition, the antibody is detectable almost immediately after exposure to the antigen. This shows that there is no lag time, which is not surprising since the immune system is already activated during previous encounter with the antigen. Antigen A can directly stimulate memory B cell to proliferate and differentiate into plasma cell and more memory B cell. These memory B cells are the ones produced during the primary immune response. Plasma cells can immediately produce antibodies, hence leads to increase in the antibody concentration in the body almost immediately after exposure to antigen A. We can see that the secondary response will provide faster response compared to the primary immune response following antigen exposure. Next, the antibody produced by the plasma cell during secondary response is from class IgG. This class of antibody is long-lived and can remain detectable for months or years inside the body after the antigen has been cleared. So, the protective value of the secondary immune response will last longer. In conclusion, the primary immune response shows how our immune system reacts to a specific antigen during the first encounter, while the secondary response is the reaction produced by our immune system during the second encounter with the same antigen. This means that every time our body encountered an antigen for the first time, it will produce primary immune response. The secondary response will only occur if our body encountered the same antigen for the second or subsequent time in the future. Since the secondary response will provide a longer lasting protection against the antigen, this is why in most cases, vaccination will require 2 doses. The first dose is to stimulate the primary immune response, The second dose will stimulate the secondary immune response, which will provide our body with a long-lasting protection against the infection.